Now let's talk about antigens and antibodies. Understanding this will help us understand the concept of transfusion. So um, let's go back to this note that we took earlier and talk about the relationship between antigen and antibodies. So once again, antigens are basically what's on the surface of your red cells, and those are determined by your genetic makeup, determined by your the kind of alleles that you have. Now, antibodies. I'm just gonna do a quick note here. Antibodies are basically proteins, and they are Y-shaped proteins that your body makes, and they are going to be found in the plasma. Now, these proteins are important for immune defense purpose, and to keep it simple, basically you have the ability to make antibodies that would target foreign antigens. Okay, so we all have antigens, and in fact, all cells have antigens. Antigens are nothing more than molecules that are found on the surface of a cell, and your antigens are going to be different from my antigens, and the bacteria's antigens are going to be different from the ones that we have. So, as a defense mechanism, your body will be able to create these Y-shaped antibodies to target any foreign antigens. That means if you see an antigen that you don't have in your own body, you are going to make an antibody to attack it. So keeping that in mind, if someone is type A blood, what kind of antibodies are they going to make? Well, if you're type A blood, then it means you will not have seen type B blood before, so you would consider the B antigen as something that's foreign. So you're going to make an antibody, a Y-shaped antibody that is going to attack the B antigen. Okay, so this is called anti-B antibody. Okay, so the key thing to remember here is you are going to make antibodies against things that you don't have. So if you're a type B person, you have B antigen, but you will be making antibodies that would attack the A antigen. Right? So this is going to be the anti-A rather, antibody. Okay, and all the antibodies, remember, they are found in the plasma. Right, the fluid portion, the 55% of your whole blood, whereas the antigen, they are found on the surface of the RBC. So maybe I should change this to antigen, and then this would be on the surface of RBC. Right? If you are type AB blood, that means you yourself have both antibody A and antigen A and antigen B. Then it doesn't make sense to create these antibodies, because otherwise you will be attacking yourself, right? So for someone who is type AB blood, they would have no anti A and no anti-B. It doesn't mean they cannot make antibodies. Like if a type AB person comes down with chickenpox or something, they can still make anti-chickenpox antibodies. But within the context of ABO blood type, they will not be making antibodies against A or B, because otherwise, again, they would start attacking their own cells, which is not good. If you're a type O person, you don't have A antigen and you don't have B antigen. So both of these would appear to be foreign to your body. So you're going to create anti-A antibodies as well as anti-B antibodies. So you will have both. Okay, so just to clarify, type O is again the absence of A and B, right? There is no such thing as O antigens, so therefore you're not going to create something like anti-O antibodies. There's no such thing. All right. With this in mind, we can now think about blood donation. Right? So let's consider transfusion. Okay? Is it safe to take blood from, say, a type A person and transfuse it into someone who has type B blood? Okay? Is this safe? As mentioned earlier, we do not transfuse whole blood. You cannot take blood from one person and then directly give it to another person. You're gonna almost most definitely harm the recipient. In order for blood transfusion to take place, you must separate the red blood cell from the plasma, and the rules for plasma compatibility are actually different than the rules for RBC transfusion. So in this course, we are only going to be looking at RBC transfusion. That means we're just going to be taking red cells, giving it from one person to another person. So here's the question, right? Is it safe to take RBC from a type A person and then give it to a type B person? Right? So in order to answer that question, there are three things you must consider. Number one, you want to ask yourself what antigens are present on the donor's RBC. You want to know what antigens are going to be found on the donor's RBC. So in this case, the donor is a type A person. So if I have to draw it out, then we are going to have some A antigen on the donor's RBC. Right? So that's easy enough. Second thing you should consider is what antibodies are present in the recipient's blood, plasma, right? So if the recipient in this case is type B blood, right, they are going to make antibody against things that they don't have, and if they're type B blood, they are going to be making anti-A antibody. Okay, so the last question you're going to ask yourself is, if you go ahead with the transfusion, will the antibody in the recipients attack the donor's RBC? Right? If you go ahead with the transfusion, and as you can see, the antibodies will recognize anything that has an A on it and they will attack it. So based on this, we know this is not safe. Okay, not safe. So type A person cannot give red cell to a type B person. Okay, and again, I cannot emphasize this enough. We are just talking about RBC transfusion. Okay, we're not giving the person the plasma or anything like that. So let's try another example. Can a AB person give blood to A person? Is it safe or not safe? So we're going to ask ourselves the three questions again. Number one, what antigens are present on the donor? So the donor is AB, which means we are going to have A antigen and B antigen on the surface. Okay, we are going to give to a type A person. So what antibodies does the type A person have? Well, if they're type A, then they would see B as foreign, and so they will have anti-B antibodies. And so if you go ahead with transfusion, the antibody will attack anything that has a B on it, and in this case, that means they will attack the red cell, and this would be not safe. Okay, let's try another one. How about taking blood from a B person and giving it to a AB person? Is this going to be safe? Well, what antigens are on the donor's RBC? It's going to be type B blood, so B antigens. If you are a type AB person, what kind of antibodies will you have? Well, remember from our chart earlier, right? AB people don't make any antibody against A or B, right? So for this, there would be none. So if there are no antibodies in the recipient, then they're not going to be attacking anything. And as a result, this is going to be a safe transfusion. Okay, so you should always kind of think about these things to see if it makes sense to go ahead with the transfusion. If you are just looking for a quick way of getting the answers without thinking through these steps, although I do encourage you to think through the steps, okay. But if you're just looking for like a shortcut, you can use this transfusion chart, okay. So type O is at the bottom, right? You might have heard of people say type O is universal donor. So they can give blood to type A, they can give blood to type AB, they can give blood to type B. Okay, and the reason they are so-called universal donor is because on the surface of a type O cell, there are no antigens, right? There's no A, there's no B. And so if you give it to these other people, it doesn't matter what kind of antibodies they have. Those antibodies are not going to react against nothing, right? So type O are the ones that can be given to all the other blood groups. Okay, but the word universal donor is not really appropriate, I think, because remember there are 30 plus blood group in the body, right? So just because you're type O for the ABO system doesn't automatically make you a compatible donor for all the other blood types, right? And we must consider all of these when it comes to a proper blood transfusion. And here, if you're AB, then you have no antibodies, no anti-A, no anti-B, which makes you the ideal candidate of receiving blood from all the other blood groups. Okay, so this is the transfusion chart for RBC. Okay, and it's kind of like a shortcut to use on the test, I guess. So once again, O can be given to A, right? And A here can be given to AB and themselves. B can give to AB and themselves, right? You can always give to other people of the same blood type. And if you want to know who you can receive from, you trace the arrow backwards. So keeping that in mind, we can come over here and look at this. We can add another column, donate RBC to, right? We can say who we can donate to, and we can say receive RBC from. So if you're a type A person, you will be able to donate RBC to another type A person, and you will also be able to donate to type AB person. Okay, you can take a look here, right? Type A can always give to themselves, and they can always give to AB as well. Similarly, if you are type B, you can give to other Bs, and you can donate to AB. If you're AB, you can only donate to other ABs. Okay, if you go back to the chart here, you can see AB does not point to anything else, right? Over here, type O, then you can give to A, you can give to B, you can give to AB, and of course, you can give to other O's. Who can you receive from? Okay, so you'll be looking at what arrow is pointing towards you. So if you're type A, you can of course receive from other A's, and the chart here, right? A, you look at what arrow is pointing towards it. So O is pointing towards it. That means you can receive from O. So similarly, type B can receive from B or O. If you're AB, then you are in luck. You can receive from all the other blood groups. And over here, if you're type O, you can only be receiving from other type O's. So this is kind of important chart to study and to understand. Don't just memorize it. Try to understand the logic behind it, and it would help you out on the test. So what does happen if you transfuse the wrong RBC? What's going to happen to the recipient? Well, there's something called cross reaction. Right? A cross reaction occurs when the antibodies in the recipient binds to the incoming RBC. So in this picture, we can see there is the type A blood of the donor, and on the surface, these little circles represent the A antigen. Right? So it's equivalent of having A's on top on the surface. Okay, and we are trying to give type A blood. In the first example, we're trying to give type A blood to another type A person. So we know this is compatible based on what we discussed, but let's review it quickly again. For type A recipient, they are going to have anti-B antibodies. So these B antibodies, they have little triangle slots at the ends, and that's not going to fit with the balls, right? Or I can put the letter B here, and because they are not the same fit, they're not compatible, then the antibodies will not bind to the incoming RBC, so nothing will happen. This would be a safe transfusion over here. So if you were to mix the blood with the antibodies in a dish, you will see the blood will mix together just fine. On the other hand, if you take blood from a type A person and you give it to a type B person, the type B person will have anti-A antibodies, and you can see the ends of the antibodies here have these round knobs which will fit into the circles here, right? The way that we have represented it so far is we actually use the letters, right? So on the surface we would have A antigens, and this type B person, they will have anti-A antibodies, which means if you mix them together, the anti-A antibodies will now bind to the antigens on the RBC. So this binding is going to cause them to clump together. Okay, so clumping together is called agglutination. All right. So in a transfusion that's not compatible, the antibodies will cross-react with the antigen. And the cross reaction will result in clumping or agglutination. And you can see in the diagram here, these big chunks, they are basically the clumping, the agglutination. And once they clump together, the next thing that happens is hemolysis. Okay, so hemolysis is the breaking. Lysis means breaking or rupture of the red cells. Okay, so if it's incompatible, we have the cross reaction, which leads to agglutination, which then leads to hemolysis. So that's not a good thing, right? Because usually when people need who need a blood transfusion, basically means they themselves don't have enough red cell, and if you transfuse them with red cells, that resulted in more red cells dying, then that's just going to not help, and in fact it's going to make it worse. So if this problem is not realized soon enough, if you transfuse like a couple bags of incompatible blood, it's possible to kill the person, and of course we do not want that. Okay, so that's what cross reaction means. So that is basically everything we need to know for the ABO system. And now we're going to talk about the Rh antigen. All right? Remember, people when they talk about the blood type, they say like AB positive or like B negative. So now we're going to talk about what the positive and negative means. That actually refers to another blood group belonging to the Rh blood group, right? And the Rh stands for the rhesus monkey, because the Rh antigen was first discovered in these type of monkeys. So let's go to the workbook. Again, we did this earlier, this question number 5 here. We filled it in for the ABO system. Now let's fill it in for the Rh blood group. Unlike the ABO system, there are only 2 alleles in the Rh blood group that determines whether or not someone has the Rh blood type or not. And the alleles are going to be the positive allele, which allows you to make the Rh antigen, and then there is the negative allele, which causes you to not be able to make the Rh antigen. So there are only 2, right? If you combine them together into genotypes, you can either be positive. Okay, you can have 2 copies of the positive allele. You can have 1 copy of the positive and 1 copy of the negative. We can usually represent it with a dash here to separate it, or you can be negative and negative. Right? So there are 3 possible genotypes for the Rh blood group. So if you have the positive copy, it doesn't matter if you have 1 copy or 2 copies, that means you are going to be able to make the Rh blood group, and that's going to make you an Rh positive person. And if you're negative, then you are going to be Rh negative person. Okay, so just to make this a little bit more clear, if you are homozygous positive or if you are heterozygous positive, right, you will be Rh positive. Okay, it doesn't matter if you have 1 copy or 2 copies, as long as you can make it, then you are considered as Rh positive. On the other hand, if you are Rh negative, that means you do not have the allele to make the Rh antigen, and that will make you Rh negative. So there are only 2 phenotypes for the Rh blood group. Now let's go through a few examples. What does it mean when someone say they are A positive? Well, that means they giving you information about two of their blood groups, right? The A is part of the ABO blood group, and the positive is part of the RH blood group. So if we were to draw their RBC, right? If they are A type A blood, then that means they will have antigen A on their surface. If they are positive, that means they will have RH on their surface. So for someone like this, what kind of antibodies are they going to have? Remember, your body will make antibodies against things that you do not have in the body. And since this person does not have the B antigen, they are going to make the anti-B antibodies. Okay? Let's try again for another person. What if you are B negative? What does B negative means? So if you were to draw the RBC again, then on the surface we will have the B antigen because of the letter B here, and negative means you do not have the RH, so I just don't need to draw it, and there will be a bunch of B's on the surface of the RBC. So what kind of antibodies are you going to make? Well, if you're a type B person, that means you have not seen type A blood before, so you will have anti-A, and here because you don't have RH, you will make anti-RH antibodies as well. Okay, the key here to remember is you make all the antibodies against all the antigens that you do not have. What if you are AB negative? AB negative. So AB negative means you will have A, you will have B, but you will not have RH. And so the only antibodies you're going to make is RH antibodies. Okay, so try to go through this yourself a few times with a few other examples, and think about what kind of antibodies you're going to make in each of the cases.